네. Okay. Hi. So I will switch to English. So yes, first I'd like to thank the organizer for inviting me here and then give me the opportunity to talk to you guys. I mean to the students and about and introducing my research area. So which is titled here as SMC complexes, which is the molecular model proteins that can shape genomes of all life form. But before that, I just wanted to give you a bit of an introduction of my group. So my group is, as stated before, that are situated in the Max Planck Institute of Biophysics, that is in the city of the Frankfurt am Main in Germany. So one of the few cities where in Germany where you can see actually the skylines, skyscrapers. So yeah, my group is started in about was 3.5 years ago. Now I have about 5 PhD students and postdoc in my lab who are all from, most of them are from very different countries. So we are very international groups. So in terms of science, and also generally speaking, what I'm interested in is to understand what would be the molecular mechanism underlying the 3D shapes of the genomes or the spatial or an dynamic organization of the genomes inside of the cells, and also how such structures can govern certain functions in genomes, which I will elaborate more detail. But I was told that, yeah, maybe it's also is kind of give you a good it's good to give you some idea about like my academia path, although it was like really nicely introduced just now. So I don't have to repeat this, but my academic path has been a bit unusual. Well, that's what I heard from other people. So because I decided to, from my master's degree, I decided to move to Europe immediately and then kind of continue to my academic careers. There were several the turning point for me that I decided to go to further and then continue my studies. Of course, I'm not going to tell all the details right now, but if you have some questions or general career advice that you are interested in studying in Europe and etc., so I just highly encourage you to contact me personally as well. Okay, going back to the introduction of my research. So the field of genome organization comes from very simple question. So it starts from very simple question. That is that how the DNA is packaged and folded inside of the tiny space of the cell's nucleus. So as you all know, it's like kind of textbook pictures of the like in the fundamental biology course. You see that if you just add up all the piece of DNA that exist within a single cells, and that will add up into the length scale of 2 meters. While if you all know that the diameter of the cell nucleus is depending on the organisms, but about 2 to 10 micrometer. 10 micrometer is in the human cells. So this obvious scale problem will give you an immediate idea about the fact that DNA has to undergo really highly compaction, high degree of compaction, just to simply fit within this such a tiny space of the cell's nucleus. So not only the fact that the DNA has to go through such a high degree of compaction, as you all know that the cells are not static objects, right? So they are going through the cell cycles. They have to go through a lot of these different dynamic processes, which involves this cell division, meaning that the chromosome inside of the cells has to go through a lot of different conformational changes. And while this conformational change is happening, also the genomes, so DNA, have to be available in the right time, in the right space, to be able to accommodate the important processes such as gene expression, replication, and also the chromosome segregation, and also the repair processes in the meantime. So such a problem, one can say, that occurs in the space-wise and also time-wise is what we are interested in to solve. And there are different types of the organization domains that exist in this like global conformation of the chromosomes. And one of this major organization domain is called chromatin loops. So what we know now is that our DNA is folded into loops, and these loops presents almost entire cellular cycle. So here in the middle, what you see is that the examples of how the chromosome conformation is changing during the different cell cycle, and if you just zoom in a microscopic level, then these different conformations or genomes are composed of these loops that are made from a single type of protein complexes called SMC. So during the mitosis, the protein complex called condensin, so they make up of these highly organized loop structures that makes up of this helical loop array, and that will be in the end made up of those X shape of chromosome that we are all well familiar with. However, when the cells now enter more into the interface regions, then these condensing gets dissociated and replaced by another SMC complexes called cohesin. And these cohesins, they are more dynamically interacting with the chromosomes and also its binding factor called CDCFs. And then by dynamically and then looping at certain regions, which will then also govern other functions such as gene expression. So I just mentioned that these loops that are present throughout the entire cellular cycle are made of a single protein complexes named as SMC complexes. So SMC is the abbreviation of the structural maintenance of chromosomes. So these are evolutionary conserved proteins, meaning they exist from bacteria to eukaryotes to higher eukaryotes like human. And these proteins contain the ATPase subunit, meaning that they can use the energy of ATP hydrolysis to be able to convert this to the more motor action. And in the eukaryotes, there are three types of protein complexes, SMC complexes named as condensin, cohesin, and SMC56. So here I show you just the schematic, brief schematic of how this protein looks like. But what you probably notice is that all these different SMC complexes, what stands out for you perhaps is that they have this global shape of the circular ring structures. So this is what the characteristic shape of these proteins. They look like a ring shape of the protein. And this ring shape of protein gets further recruits further additional the protein complexes, which can be dependent on the specific SMC complexes. But what these protein does is that they can stimulate the loading of these proteins onto the your gene and the chromatin, and also stimulate the ATPase activity. So what I told you so far is that this throughout all different cellular cycle, there has been always the loop, chromatin loops present that is formed by these SMC complexes. And in this field of the SMC and all in general chromosome biology, one question that has been puzzling us for a long time was that how these SMC complexes can make loops. So there has been two competing theories or hypotheses about this. So the one theory was that these SMC complexes could make loops by just passive random DNA clone linking, meaning that they are just acting as a passive linker. So they just kind of, when there is genomes are already folded in a very tiny space of nucleus of cell, then it can just zip the nearby regions in the, and then therefore can form the loop. The alternative theory, however, was that this was the hypothesis called DNA loop extrusion. So that is that this ring-shaped protein, since it can hydrolyze the ATP, it can use the energy of ATP and then transfer that through certain conformational changes to capture a small part of loop already existing in the genome. But through the burning and then hydrolyzing ATP, it can sort of gradually reel the DNA and into the shape of the loop. So that's more is coming. So then that gives the idea about how SMC complexes can act as a motor protein that can actively generate the loops. So between these two competing hypotheses, the recent series of the single molecule experiments, which are partly from my own work during my postdoc period, as well as the recent work from my own group and also other work regarding coesin. So we have, these work have confirmed now that SMC complexes can indeed work as a motor protein that can actively extrude DNA into the loops. So now, of course, the question is, how did we actually verify this hypothesis, right? So the way we verify hypothesis was to directly seeing these processes with our eyes. So for this, we used so-called in vitro single molecule visualization technique to monitor in real time this active DNA loop extrusion process that is performed by the different SMC complexes. So this is done by using the one type of fluorescence microscope called total interreflection wide field fluorescence microscope. So that is not too different from any type of fluorescence microscope you might have experienced during your lab courses. So this under this microscope, we combine that with our home-built flow cells, where we use the DNA, the bare DNA, as a substrate to visualize this the loop formation. And this DNA has a length about 50 kilobase pairs, or like 16 micrometer in length, and their ends of the DNA are tethered on the flow cell, and we stain them with the dye, intercalating dye, and also subsequently inject the SMC complexes that is labeled with the fluorophore. So then what you see, that if you now look at the movie that I'm playing, so there is the piece of DNA that is just fluctuating, you see in the middle, while both ends of the DNA gets now, it's not moving so much because they are tethered into the end, on these ends. So these are the DNA. And if the just protein is not doing anything and then they are just mainly bound on the DNA, then you see this co-fluctuation. So you know that this protein is now bound on the DNA. So this just very direct imaging allow us to understand the interaction between the DNA and these SMC proteins. So what we did further to visualize the real-time this loop extrusion is by adding applying the constant buffer flow during the loop extrusion process. So you can imagine the DNA is very flexible, polymer-like project. The property, it has properties of that. So meaning that if you just further stretch them by just applying tension that is done here by the buffer flow, this DNA will be further stretched. That's why now you see the DNA has a shape of the like arc shapes instead of this like linear shape. And then therefore you can gain the resolution a bit better. And then you can better visualize this change of the conformations that are happening during the loop extrusion processes. So using this method that we have, for example, showed recently in the case of SMC56 that these proteins can indeed work as a DNA loop extruder. So what you see here now is the image of the DNA only. The protein is not labeled, but this initial arc shape of DNA, you will see that now there's a like more concentrated puncta that is formed in the middle, and then which gradually changes the shape into the loops as the time goes by. So that is driven by the protein that is bound at the stem of the loop. And to confirm that these are indeed performed by the protein that is bound at the stem of the loop, one can co-visualize the DNA, which is in the left side, and the protein that is labeled with a different color that's in the middle, and then you can combine these images, which is on the right side. Then what you see here now that again, the conformation of DNA is changing into the loops. So you see this puncta is the kind of gets elongated, so that is the loops, while the protein is consistently located at the stem of the loops. So this direct visualization of the loop formation processes, and then the fact that the loop was always started as a very small puncta, but gradually increasing its length, that directly confirmed that this these proteins are indeed the active motor proteins that actually can generate the loops by the extrusion processes. So, so here I give you the example and example video of how we really can visualize this single molecular interaction processes between the SMC motor proteins and DNA. And for the rest of the talk, I will give you a brief introduction of the two papers that has been recently published in my lab. So that is first is about understanding the motor activity of SMC56 as a DNA Lubbock string motor. And the second one will be about another SMC complexes, in this case a bacterial SMC called Watt's complex, and how this Lubbock extrusion activity of the bacterial SMC have actual additional function, which is to cleave the DNA specifically. That is acting against, that is for defending the host genome against the virus DNA, viral plasmids. So let me again go back to the SMC56 complex. So this is again one of the SMC complexes that are present in all eukaryotes. So the function of SMC56 has been much less characterized as compared to condensin and coesin complexes, but their function so far, what is known that they are involved in maintaining genome during or in preparation of the DNA replication, and also during the repair processes and the chromosome segregation. And also, the more recently identified role of this SMC56 complex is that they actually can work as an antiviral factor which defends the human genome against the viral DNA infection. In terms of structure, as we are always interested in, as a molecular biology, we are always interested in structures and their functions and mechanisms. So what stands out in the case of SMC56 is that they actually have additional subunits that are not shared by any other SMC complexes. So that is NSC2, which work as a sumo ligase, and then NSC56, which has been thought before as working as a DNA loader complex. So now I showed, just before that, I showed you the two example movies of how we confirm the DNA loop extrusion activity, but both movies were recorded while we are applying the buffer flow, constant buffer flow, to visualize this lengthening process of the loop. But what we actually also can do is that we just monitor these loop extrusion processes without any buffer flow. So then what you see, loop formation as this puncta that is generated in the midst of the DNA, and then that increases the length, meaning that there are more and more dyes accumulating the loops. And therefore, you see this higher intensity of the punctum in the midst of the DNA. Until up to the point there is a protein gets dissociated, therefore the loop disappears. So what you see in the middle, this is called kymograph, meaning that in each snapshot of the movie is now summarized into the one line of these images. So that is by summing up all the fluorescence intensity along the length of the DNA. And from there, we can actually get more quantitative information, such as like what is the length of the DNA loop that we have at the different timeline, and what is the DNA left over outside of the loops. So from there, we can get, for example, this kinetic graph of the DNA size changes over time, which you can see in the right bottom figures. And this kind of DNA kinetic graph will tell you actually more interesting and also quantitative information about, for example, what is the speed of the DNA loop extrusion and whether this loop extrusion happens in two-sided or one-sided manner, or what's actually the force that this protein stops its working. So the speed of the loop extrusion we found is that they actually are very fast motor, meaning that they can extrude the loops into the speed of 1 kilobase per second. So that is a very, very fast motor as compared to any other motors that we know. for example, like RNA polymerases or helicases and so forth. In terms of symmetry of the loop extrusion, meaning whether during the growth, whether this loop is consumed by only one side of the loop or from both sides, we found out that in the case of SMC56, they are symmetric motor. And the stalling force of this SMC56, they are a very weak motor, meaning it's a very small force of only 0.4 piconewton that they actually stops their workings. What we also found out in the case of SMC56 is actually the dimerization of these two complexes are required to start or initiate the loop extrusion. So this estimation of protein stoichiometry can be done because we again label our proteins with a single dye, single fluorophore, meaning that and this single dyes have this the characteristic of that like photobleaching, meaning that if you shine the single dye with the extended lifetime, then they will bleach out and then they will never come back their signal again. So by just simply monitoring this intensity of this fluorophore and then counting that how many bleaching steps, the steps downwards that you see during the extrusion, you can actually count how many molecules were there. And what we observed was the majority of this loop extruding event was accompanied with this two stepwise photobleaching step, suggesting that these SMC56 are working as a dimers. What we also found out was that although during the extrusion, these SMC56 are working as a dimers, there are the cases there are single SMC56 are still interacting with DNA. So in this case, what SMC56 does is that they just lands on a DNA here. You see this red dot that is just moving unidirectionally along the length of the DNA. And this process is again ATP dependent. And once the dots, so this protein, single protein, is landed on the ends of the DNA, then they just stably remain bound in these ends until that they bleach out in a single step. And then the speed of this translocation is about a similar speed as the loop extrusion that we observed. What we also observed was that when the single complexes, they bind and then start translocating, and then when the second protein comes in and then they binds at the same location where the initial single complexes were there, this is when the loop extrusion started. So that's the direct observation of the translocating single SMC complexes can transform into the loop extruding dimers. So what we also found out in this particular paper was that we have shown that this NSC56 subunits, which was not very well characterized before about their exact function, was that they actually can inhibit the loop extrusion process of this SMC56. Because when we delete these two subunits from the rest of the complexes, we observe actually great stimulation of the loop extrusion activity. However, when we compare that the loop extrusion speed of this SMC56 in the presence of NSA56 or not, and also the lifetime of the loop extrusion in the presence of an NSA56 or not, we observed that these both loop extrusion rate and the loop dwell time was similar, suggesting this NSA56 mediated loop extrusion inhibition happens only at the start of the loop extrusion. So, and then we were questioning basically what would happen for SMC5, so for NSC56 to be able to inhibit the loop initiation process of SMC56. And for us it was logical to think that, okay, so for SMC56 to be able to extrude loops, so two processes has to be done. So first they have to be loaded on the DNA, and then second they have to be able to dimerize. And we were wondering whether NSC56 can indeed inhibit the dimerization process of the loop extrusion. So to address these questions, we turn back, we decided to use the technique called mass photometry. So that is the scattering-based imaging, where you can actually count, measure the molecular weight of the single protein that is randomly stuck on those, your glass slides of the surface. And from there, what we observed in the presence of DNA, and SMC56 actually present in both monomeric and the dimeric form, while in the presence of NSC56, this dimer fraction disappears, gray or significantly reduced, and then instead there is additional peak appears that is basically a single SMC56 complex where the NSC56 is the bound on it. So with this was a direct, the visualization of that NSC56 association inhibits the dimerization process of SMC56. And what we also observed was that this, while NSC56 can enhance the DNA translocation activity, inhibits the loop extrusion activity, it also at the same time can enhance the DNA translocation activity, which is again majorly done by the single complexes. So in the presence of NSC56, we observed that these proteins don't really extrude loops on DNA anymore, but simply they are mostly just translocated along the length of the DNA. So with all these different observations or different findings combined, we finally showed the initial model of the loop extrusion mediated by SMC56. So there is SMC56 when they in the presence of ATP and DNA. When they are in a single complex, they can translocate along the DNA. While they are in a dimeric form, they can initiate the loop extrusion. And this dimerization can happen either when these already dimer complexes can land on the DNA and initiate loop, or there's a single translocating complexes, further recruits the additional one, and then the loop extrusion starts. And the role of NSC56, so the subunit of this SMC56, is that it inhibits the loop extrusion processes or it regulates it by inhibiting the dimerization. So so now I will start introducing the second work, which is about, again, another SMC56 complex, but it exists in bacteria. So I started my talk by mentioning basically what are the functions of SMC complexes. And although many SMC complexes are known their role of organizing genomes into the loops, the more recently discovered role of certain SMC complexes they are actually working as antiviral factors. In the case of human SMC56 complex actually can inhibit the transcription or silence the transcription of the broad spectrum of viruses, and which has the particular topology of this circular DNA. And their activity is dependent on this the loader called SLF12. Similar role has been identified in the case of bacteria, or 10% of bacteria. So that is the bacteria SMC complex called WATjet. So what these WATjet complexes are doing, so when they have, they can specifically cleave, so the cut off these the circular plasmids that are the phage DNA, with the help of these motor complexes, together with its own specific subunit ZD, so that is similar to topoisomerase 4, which can cleave the circular DNA. So in 2022, there has been 2 papers published back-to-back, which shows that, demonstrated indeed this WATjet complexes can specifically cleave the circular topology of the DNA as a function to defend the host genome against the phage DNA. And these 2 papers were suggesting that maybe this loop extrusion activity of this WATjet, which was again not known before, might be the underlying mechanism of the plasmid cleavage activity of WATjet. And this is because of the structural similarity of the WAZ to other loop extruding SMCs, and also it might be, and their plasmid cleavage activity of WAZ is dependent on the ATP, but not depending on the DNA sequences, not like this typical nuclease activity containing enzymes. But their activity is also topology specific, meaning they only can cleave circular DNA, but not linear DNA. So we decided to look at, again with our visualization technique, the workings of these WAZ complexes in our flow cell. And then we first starting with the linear DNA. And then we indeed first confirm the DNA loop extrusion activity of WAZ complex. So here in this case, you see another loop extrusion movie where the DNA puncta is transforming into the loops, and then during the time that the protein, WAZ protein in this case, is stably located in the stem of the loop. So now the movie again plays. Yeah, so here you see the binding of this protein that kind of transforming into transforms the DNA conformation into the loops. So this was the first direct evidence of the loop extrusion activity by any prokaryotic SMC complexes. So in this case, similar to SMC56, we also observed was that this protein, they can actually extrude loops as a dimers. Again, confirmed by the photobleaching step characteristics, where during the extrusion, we see two consequent stepwise photobleaching steps. And because these were dimers and then extruding loops, then one can also imagine that this extrusion happens from the both sides of loops that acting where individual proteins independently extrude loops. So indeed that was the case, because during the growth of the loops, which you can see in this kinetic graph, both sides of DNA, so the DNA upside, let's say, and then downsides, they both decrease the length of the DNA. And then we went a bit further and then asked the question about whether the dimerization of these two WAZ complexes are really required for the loop extrusion activity. So what we did was that we decided to partially truncate the ZA domain, one of the subunits of this WAZ, so which is in this that I'm indicating now, and so that we made two different constructs, which is absent of the rest of the binding domain of JET-B and C, or JET-C only, meaning that after assembling all these wild-type complexes with the truncated complexes, then we will end up with the stoichiometry that is corresponding to the monomeric complexes. So as compared to the wild type, which we observed around 80% of dimeric complexes, in these artificially monomerized complexes, we observe the majority of protein are existing as monomeric complexes. And then what we observed was that these artificially made monomeric proteins were unable to extrude loops, which is shown in this bar graph. In both cases of monomeric complexes 1 and 2, we observed a significant reduction of the loop extrusion event. And when we actually zoom in, very unlikely event of the loop extrusion that happened in these monomeric complexes, we observed that these minor events were also coming from the dimers, that is coming from this very small fraction of dimer complexes in this case. So indeed, we confirm with these truncation experiments that dimerization is indeed required for the loop extrusion activity of WAZ. And then, as I mentioned in the beginning of this introduction of this work, that the hypothesis in the field was that the loop extrusion activity of WAZ may be required for the DNA cleavage activity of the WAZ, that is specific to the circular DNA. So we decided, of course, to understand that we need to change the topology of DNA, so not like real working with linear DNA only, but also with the circular DNA. So we modified our flow cell so that we can specifically label the plasmid DNA onto our flow cell. So the circular DNA you will see, for example, in this left side of images, so these individual stretches that you see are the circular DNA that is stretched on with the constant buffer flow. If you compare that with the linear DNA, which has the same length, you immediately notice what is linear and what is circular, because circular DNA simply has like half of the length of the linear DNA, and also their intensity is like about twice. So after developing a assay where we can immobilize circular DNA, we first looked at how this Watson complex can extrude loops on this circular DNA. So here in this movie, what you observe is the summary of this observation of the loop extrusion that is mediated by Watson on a circular DNA. So what happens is basically these proteins, they like to bind near the tip of this the banded structure of the circular DNA, and then through the extrusion they can sort of move along the length of the plasmid. And which you also can see in this chymograph. And what we observe is that through the extrusion, these proteins gets more and more. So first, they initially bind in somewhere in the circular DNA, and then they end up in the anchor tethered anchor point of the circular DNA. And so over time, more and more proteins gets accumulated near the surface of this the flow cell, and therefore more and more proteins are now situated at this near the surface point. And in contrast, what we observe in the case of linear DNA, that these proteins, they bind somewhere in the middle of the linear DNA, and then they extrude loops. And then by doing so, when they meet this free end of this linear DNA, they actually disrupt loops, and then they get dissociated. So therefore, the circular DNA, we observe way more accumulation of the proteins as compared to the linear DNA. And in the case of circular DNA, we indeed could observe the specific cleavage activity of Watson. So here what you see is the larger field of your images. Left side is DNA channel only, right side is the DNA with the SMC complexes. So here I show you this grid boxes, where and then with the numbers, so where the cleavage activity will happen in this following order. So now I show you the video. So first, this plasmid, the DNA over time now gets linearized. And the second one, the linearization event happens just now. And the third one, yes, now happening, fourth, fifth, and then sixth. So I hope you catch some of these events. But if not, that I can play, at least zoom in one of the events, which is the basic summary of this paper. So what we observe is the following. We observe that this protein actually, they binds and then they extrude loops on the circular DNA. And then only when they reach these near the surface end of this flow cell, and then where they changes actually conformation, then that's when their plasmid cleavage activity gets activated. Therefore, they can cleave the DNA. And we observe that nearly 100% of cleavage event happens at least one full length of the loop extrusion, which really shows that cleavage activity cleavage activity requires of its loop extrusion activity. So after these experiments, we could provide a summary, summary model, which is basically that the Watson complexes, they can extrude loops in both linear and circular DNA. However, in the case of linear DNA, once through the extrusion, they reach this open end, they will just get dissociated, and then therefore they cannot activate their cleavage function. While in the case of the circular DNA, after full length of extrusion, they can change their conformation into the stuck state or stalling state, which then their nuclease activity function gets activated. Therefore, the DNA cleavage activity happens. So with that, we'll conclude my talk or lecture. And yeah, so if you have any questions, so please feel free to ask. Thank you.
네, 교수님, 발표 감사드립니다. 네,
네.
네, 되게 재밌게 잘 들었습니다, 교수님. 이게 몰레큘러 이미징 같은 거죠? 이렇게 보면 이렇게 단분
아, 네.
몰레큘러 이미징을 보면 된 거죠?
네.
되게 엄청 신기하네요. 네, 왜 비슷한 연구를 하시는 분이 계셔가지고 비슷하게 뭐 이렇게 분자끼리 서로, 서로 사이에 그 프레시 작용을 측정한 걸로 이렇게 분자 거리를 측정한 거 봤는데 이렇게 DNA랑 단백질을 이렇게 형태학적으로 보는 건 되게 신기한 것 같습니다. 네. 네, 혹시 학생들이 질문을 할 건데요. 그 혹시
네.
한국 학생들은 우리말로 보통 질문 많이 하고요. 그 외국
네.
친구는 아마 영어로 질문할 것 같아요. 교수님께서는 편하게 대답해 주시면 됩니다. 네.
네, 알겠습니다.
여기 조민영 학생 한 명이 질문이 있는데요. 조민영 학생 질문하셔도 돼요.
들리시나요?
네, 잘
네,
들립니다.
잘 들려요. 네.
교수님, 제가 간단한 질문 하나 드리고 싶은데, 혹시 SMC 몰레큘이
네.
DNA를 인식하는 특정한 메커니즘이 있을까요?
아, 그, SMC는 DNA recognition을 하는 게 현재까지는 특별한 메커니즘은 없고요. 그러니까 어떤 엔자임들은 DNA sequence dependent한 뭐 바인딩 액티비티를 보인다든가 하는데 SMC는 그런 건 없고 또 하지만 우리가 현재 알고 있는 거는 만약에 그 DNA 시퀀스가 AT 시퀀스가 굉장히 리치하다. 그거는 이제 DNA가 그 훨씬 더 플렉서블하다는 얘기거든요. 근데 그럴 때는 SMC가 더 잘 바인딩이 되는 거는 알고 있습니다.
감사합니다.
네, 다음 이도원 학생 질문하셔도 돼요.
안녕하세요. 혹시 들리시나요?
네,
네,
저는 그 SMC5, 6 컴플렉스가 루프 익스트루전 속도 효율이 코에신과 콘덴신과 어떻게 비교되는지 혹시
네.
ATP 농도나 아니면 환경 조건에 따라서 다르게 뭔가 선택이 되는 건지 아니면 같이 작용하기도 하는지 코에신이랑 콘덴신이랑 그거
예. 그 루빅 슈전 스피드는 지금까지 현재 그 인비트로 익스페리먼트에서 캐릭터라이제이션이 된 거는 SMC5, 6와 코이신 그리고 컨덴신 그 셋 다 거의 다 비슷한 속도를 보이고 있거든요. 그러니까 예. 그리고 ATP 하이드로시스도 그 뭐지 속도도 다 비슷하고요.
네.
아, 코에신하고 SMC5, 6는 그 셀 사이클에서 셀 사이클에서 같이 펑션을 하기 때문에 두 개는 뭔가 인터랙션을 할 것으로 지금 예상은 하는데 물론 그거에 뭐 다이렉트한 뭐 증거가 있는 건 아니지만 컨덴신은 셀 사이클에서 마이토시스로 셀이 넘어간 다음에 그 다음에 작동을 하기 때문에 그 뭐지 시기가 다르게 작동을 하는 걸로 알고 있습니다.
혹시 또 한 개 질문 드려도 될까요?
네,
그 유캐리오틱 생물에서 보편적으로 발견된다고 하셨는데
네,
혹시 그 다양한 생물이 있잖아요. 종에서 동일하게
네.
작용을 하는 건지 아니면 종 특이적으로 SMC 컴플렉스가 좀 작용을 하는 건지 그것도 궁금하거든요.
그러니까 물론 우리가 모든 그 오르가니즘을 다 테스트를 한 건 아니지만 그 SMC 그 관련된 그 SMC5, 6와 그에 관련된 쥐는 다 현재 모든 유카리오트에서 보이고 있고요.
네,
근데 물론 그 펑션 그 각각의 오르가니즘 그 펑션이 뭐냐 이제 그거는 좀 더 인비보 익스페리먼트가 생물학 그런 실험이 더 이제 많이 증명이 되면 그때는 좀 더 그 알겠죠, 그 롤을. 예.
감사합니다.
네.
네, 다음 이소영 학생 질문하셔도 돼요.
들리시나요, 교수님?
네,
네, 들립니다.
발표 감사합니다. 일단,
네.
루빅 스튜전이 어떤, 그 DNA 어떤 도메인에서 많이 나타나는지 아무래도 좀 패턴 같은 게 있을 것 같아서 이게 좀 궁금하고요. 그리고
네.
루비 형성됐을 때, 이게 플렉시블하게 그 존재를 하는지 아니면은 그 리지드하게 그 폼을 그 유지를 하는지를 궁금합니다.
네. 아, 첫 번째 질문이 DNA 어떤 도메인에서 루빅 슈즈를 발현을 하는지였잖아요. 그거는 SMC 컴플렉스마다 다른데 예를 들어서 코에이션을 보면 우선 SMC는 룹을 어떤 DNA 도메인이든지 만들 수 있거든요. 근데 그 대신에 어떤 그 도메인 스페시픽한 롤은 SMC가 그것을 주는 것이 아니라 그 SMC랑 그 인터랙션 하는 다른 프로틴이 줍니다. 예를 들어서 CTCF 같은 프로틴은 코이신이랑 스페시픽한 인터랙션을 하거든요. 그래서 그 코이신과 그러니까 예를 들어서 코이신이 룹을 만들고 처음에 룹을 만들면서 그 CTCF를 만나면 거기서 룹이 스탑이 됩니다. 그래서 그 룹이 스탑이 되는 그 룹 도메인은 아, 그 셀 에버리징, 뭐 지놈 시퀀싱을 해도 그 도메인은 똑같이 보이거든요. 그래서 그런 룹 도메인이 스페시픽하게 돼 있고요. 근데 그런 룹 도메인은 예를 들어서 진 익스프레션이든가 아니면 뭐 DNA 리페어든가 그런 어떤 다른 그 지놈 펑션과 관련이 있는 더 근접하게 연결이 있다고 알려져 있습니다. 그리고 두 번째 질문은 룹이 형성이 어떻게 두 번째 질문이 뭐였죠?
그러니까 룹이 형성이 되면은 이게 모양이 자유자재로 바뀌는 건지 아니면 그 형태를 계속 유지하고 있는 건지 궁금합니다.
예. DNA는 플렉시블하기 때문에 물론 그 룹이 도메인이 형성이 돼 있어도 이제 당연히 그 룹 도메인 자체는 플렉서블 하겠지만 그 인비보에서는 당연히 그 스페이스가 굉장히 적기 때문에 물론 룹 도메인이 형성이 돼 있으면 그 룹 안에서는 뭔가 그 안에서 일어날 수 있는 다른 뭐 메커니즘들이 더 자주 일어나는 그런 타드 도메인이라는 게 형성이 되고요. 그 룹 바깥에서는 또 그 룹 바깥에서 일어날 수 있는 그 다른 도메인이 형성이 되기 때문에 우리가 지금 현재 알고 있는 거는 룹이 만들어진 그 도메인에서는 그 지놈에서는 더 서로 인터랙션이 많이 일어나고 룹 바깥 도메인에서는 그 셀프 인터랙션이 더 많이 일어나고 그렇게 알고 있습니다.
네, 감사합니다.
네.
네, 다음 박검수 학생 질문하셔도 돼요. 질문이 엄청 많네요.
좋은 강연 감사합니다. 그
네.
SMC5, 6 컴플렉스랑 그 DNA 움직임 차이를 보여주는 영상에서 그
네.
DNA 앞부분을 고정한 걸로 저는 봤는데
네,
그
맞아요.
방법이 좀 궁금합니다.
그 DNA 끝 쪽을 바이오틴으로 레이블이 돼 있거든요. 그래서 저희 플로우셀 서피스에는 스트랩타빈이 있어요. 그래서 바이오틴하고 스트랩타빈 그 바인딩을 이용해서 그 DNA를 고정을 예, 고정을 했습니다.
아, 네, 감사합니다.
네.
네, 다음 이상희 학생 질문해 주세요.
네, 좋은 강연 감사합니다. 혹시
네.
최근에는 SMC를 타겟으로 하는 어떤 종류의 뭐 약물 개발이나 치료적 접근법이 밝혀지고 있는지 궁금합니다.
SMC는 좀 더 아직은 기초 연구를 좀 더 많이 되고 있는 추세이고요. 그리고 물론 그 뮤테이션, 뭐 코에신 같은 경우는 그 어떤 그 SMC 그 서브 유닛 중에 하나의 뮤테이션이 됐을 때 어떤 뭐 뉴로제네틱 디지즈든가 아니면은 뭐 디벨롭먼트 디지즈 뭐 다운 증후군이든지 그런 게 발현이 되는 걸로 알고 있기 때문에 또 캔서와도 발현과 연관이 있는 걸로도 알기 때문에 그쪽으로도 당연히 연구가 되고 있긴 하지만 좀 아직은 좀 더 기초 단계라고 할 수 있고요. 그 대신에 이제 그 페이션스에서 발견된 그런 그 뮤테이션 자체를 우리 몰레큘러 바이올로지에서 아티피셜하게 그거를 뮤테이션 넣은 다음에 그게 룹 익스트루전 그 액티비티에 어떤 그 영향을 미치는지 이제 그런 연구를 그런 페이퍼가 나온 적은 있습니다.
감사합니다.
네.
네, 다음 이예빈 학생.
네, 교수님 좋은 강연 잘 들었습니다.
네.
그 SMC5, 6에 존재하는 그 네거티브 레귤레이션 서브 유닛 그 NSE 같은 것들이 그 SMC와 비슷한 역할을 하는 엔자임들에도 존재하는지가 궁금합니다.
네, 네, 감사드립니다. 예, 아, 물론 아주 그 정확한 메커니즘은 다를 수 있지만, 예, 그 SMC 필드에 지금 굉장히 또 중요한 문제, 어떤 퀘스천 중에 하나는 이게 어떻게 레귤레이션 메커니즘이 지금 어떤가를 가 굉장히 토픽 중에 중요한 토픽 중에 하나인데요. 네, 지금 질문하셨던 것처럼 예를 들어서 NSE 56처럼 예를 들어서 코에이션 SMC 몰라큘에서는 와플이나 아니면 뭐 PDS5 같은 다른 그런 섭 유닛들이 그 코에이션이 룹을 만들면 그 코에이션이 액티브하게 그 디스트럭션 하는 그 DNA에서 디소시에이션 하는 것을 중재한다든가 아니면 그렇게 해서 그 룹을 좀 더 다이나믹하게 만든다든가 아니면 이미 그 룹 형성된 룹 도메인을 그러니까 없애는 역할을 한다든가 그런 것들 그런 많은 코팩터들이 현재 발견이 되고 있습니다.
감사합니다.
네.
네, 다음 한지 학생.
네, 강의 감사드립니다, 교수님. 최근에 우주의학에 대한 관심도가 좀 증가하면서 미세 중력의 영향에 대해서 좀 다양한 연구가 진행되고 있다고 알고 있는데요. 교수님께서 연구하시는 그 모토 프로틴의 경우는 좀 중력의 영향을 많이 받는 분야일 것 같아서 혹시 그런 방향으로 연구가 되어 있는지 좀 궁금합니다.
물론 제가 그 분야를 잘 알진 않지만 제가 알고 이 모든 것들이 나노 스케일에서 일어나기 때문에 물론 그런 정말 이 나노 스케일에서 그 중력의 영향이 얼마나 큰가는 잘 모르겠지만 프로틴의 액티비티는 똑같을 것 같아요. 하지만 이제 DNA의 퀘스천으로 가면 또 다르겠죠. 왜냐하면 DNA 그 나노스케일 프로틴이 그 다루어야 하는 DNA는 사실 훨씬 더 큰 마크로 스케일이니까 그럴 때는 중력의 영향이 있을 수도 있겠죠. 네,
감사합니다.
네,
네, 다음 지준환 학생.
네, 교수님, 좋은 강연 잘 들었습니다.
네,
질문이 두 가지가 있는데, 첫 번째는 이제 원래 전공이 물리학 쪽 분야셨는데 어떻게 생물학 쪽에서도 이제 분자 생물학 쪽에 관심을 가지게 된 계기가
네,
궁금하고요. 두 번째로는 저는 이제 스템셀을 다루고 있는데,
네.
SMC 컴플렉스를 스템셀에 부여를 해준다면 혹시 스템셀이 디퍼런시에이션이나 매추레이션에 영향을 줄 수 있을지, 또는 이제 관련 연구를 진행하신 적이 있으셨는지가 궁금합니다.
네, 감사합니다. 우선 첫 번째 질문, 저는 좀 전공을 많이 바꾼 케이스인데요. 처음에 이제 학사는 공학으로 하고, 그리고 물리학으로 갔다가, 그리고 이제 좀 더 지금은 몰라큘러 바이올로지랑 비슷한 연구를 하는데, 이렇게 급격하게 연구를 바꿨다기보다는 예를 들어 제가 물리학에서 박사학위를 받을 때 그때도 이미 생물 물리랑 비슷한 연구를 하고 있었기 때문에 그때 처음 이제 바이올로지 필드에 접근을 하게 됐고 거기서 이제 바이올로지 어떤 그런 그 하이포테시스가 있고 그것을 어떻게 어떤 메소드를 이용해서 어떤 그것을 밝히냐 뭐 이런 것들에 어떤 되게 정말 연구의 즐거움을 느꼈기 때문에 조금 더 지금 물리학 몰라큘러 바이올로지 쪽으로 간 것이고요. 그러니까 좀 더 트랜지션이 그러니까 그냥 자연스러웠던 것 같아요. 연구 그 당시에 했던 프로젝트에 그거에 관련이 되어서 조금 자연스럽게 트랜지션을 한 경우이고요. 그리고 스템셀 디퍼런시에이션에 대한 그 SMC의 관련은 사실 그 분야는 저랑 굉장히 좀 멀기 때문에 제가 뭐 물론 당연히 그 분야에서 연구를 한 적은 없고 아 이건 뭐 다른 얘기지만 좀 다른 분야이긴 하지만 현재 또 지금 많이 연구가 되고 있는 분야는 그 어떤 그 코에신이 만든 그 룹 도메인이 어떻게 어떤 뭐 뉴로제네시스에서 그런 그 다른 뉴런 셀들이 이렇게 그 디퍼런시에이션을 할 때, 근데 그리고 그 그게 이제 진 익스프레션하고 관련이 돼 있는 거잖아요. 근데 그런 다른 디퍼런시에이션에 관련된 진 익스프레션이 어떻게 그 코에션 룹 도메인과 관련이 있는 거 이제 그런 연구들은 많이 진행이 되는 걸로 알고 있습니다.
네, 감사드립니다.
네.
Next, Joya, is your turn. You can ask questions.
Yes, thank you so much for your presentation.
Yeah,
Unfortunately, my major a little bit far away from your topic. So the question is, in the beginning of your presentation, you mentioned about three main complexes as CMS56 complex, cohesine, and condensine, right?
Mm-hmm.
And I'm just wondering about the last two. They are also involved in the forming of DNA loops.
The last one, yes, the last one I didn't really mention because of this. Okay, so the SMC complexes, so they exist in all domains of life. So meaning they also exist in eukaryotes and also bacteria. So these three complexes that I mentioned initially, so that are present in all eukaryotes. But the ones that I mentioned later, the Wattjet complexes, these are bacterial SMC complexes. Yeah.
Thank you.